Dear students, welcome back. In the previous session, we have discussed about SD curves. Now, in this session, we are going to discuss about electromyography. Okay, electromyography is the study of motor unit activity. It involves detecting, amplifying and displaying the electrical changes that occur when a muscle contracts. It provides a powerful tool for documenting the role of muscle in physical activity and for assessing the integrity of the neuromuscular system. An EMG examination assesses the integrity of the neuromuscular system, including the upper and lower motor neurons, the neuromuscular junction, and muscle fibers. The test usually involves observation of muscle action potentials from several muscles in different stages of muscle contraction. And also, this test should be integrated with other methods of assessment such as assessment of muscle strength, sensory test, and test for reflexes to come to a clinical conclusion. Okay, so before understanding EMG, one should have the knowledge about the motor unit and the mechanism of conduction of impulses through the nerve. So normally the motor units consist of an anterior horn cell. So this one is present in the anterior horn of the gray matter of the spinal cord. Anterior horn cell and one axon, only one axon. So one neuron and its axon and its neuromuscular junctions and all the muscle fibers innervated by that axon. This is called as motor unit. Okay, when an electrical impulse is given, muscle contraction is produced. By repeated depolarization and repolarization, the impulse is transmitted through the motor units. And once the impulse reaches the muscle membrane is depolarized at the same time, which is producing muscle contraction. So, during this process, the muscle membrane is depolarized. As it is depolarized, it produces some electrical activity that is manifested as one motor unit action potential. One motor unit action potential. That one is simply called as motor unit, MUAP. MUAP is called as motor unit action potential. Okay, and this one is recorded graphically as EMG. Okay, so EMG is electromyograph here. Okay, though the signals given are very tiny of a few microvolts, so they are very few microvolts, and they are amplified to 1,000 times to give these microvolt values that can be displayed an oscilloscope. Okay. Operate a loudspeaker. So this oscilloscope is connected with a loudspeaker, producing some characteristic sounds and can be recorded on a chart. Okay. So now this is the process how these electrical impulses are obtained through EMG. So normally this recording of this EMG requires 3 phases. One is an input phase, another one is a processor phase, another one is processor phase, and the last one is, third one is the output phase. Okay, an input phase includes electrodes. It includes electrodes to pick electrical potential from contracting muscles, and the processor phase during which small electrical signals is amplified, and an output phase where the electrical signal is converted into visual or auditory signals, okay, so that the data can be displayed and analyzed. Okay, so this is about the EMG recording system. So if you see the recording electrodes, normally recording is made through a coaxial needle electrode. Normally diagnostic EMG cannot be carried out with surface electrodes. Okay, so and this, what happens means, once when a normal muscle contracts, all the muscle fibers in the motor unit depolarize, at the same time causing a local electrical disturbances, okay, in the muscle. This disturbance produce myoelectric signal. So that can be detected either by electrodes placed on the skin or by the needle electrodes. An electrode is a transducer, a device converting one form of energy into the other. It can be a monopolar or bipolar in the arrangement. For any number of here the electrodes may be of different types are there, but EMG purposes where single motor unit potentials can be recorded from different parts of the muscle, whether the needle electrodes are used or maybe the other type of electrodes. Okay, so the electrical disturbances recorded is the sum of the potential due to all the muscle fibers in the motor units. So for any study, the number of electrodes required here is three electrodes. Okay, it may be a needle electrodes. If you take needle electrodes only, so that one also, here we required 3 electrodes. One is the recording electrode, another one is a reference electrode, and next one is the ground electrode. These are the 3 types of electrodes. If it is a monopolar technique, the electrode placement is like that. The recording electrode is placed over the muscle belly. So for example, this is the muscle belly. So one electrode is placed on the muscle belly, and the second electrode, that is surface or reference electrode, is placed over the skin near the site of insertion. So reference is at the belly of the muscle, so that is recording electrode, and reference electrode you have to kept at the insertion. Ground electrode is also placed nearer to the recording electrode, but not over this thing. So what is the function of this ground electrode means? This ground electrode provides a mechanism for cancelling out the interference effects of external electrical noises. So it will cancel the external electrical noises. Okay. So once this myoelectrical signal, which is produced from the result of the muscle or nerve depolarization, so which is converted into an electrical potential, that one is capable of being processed by the amplifier. Okay, so it is the difference in the electrical potential between the two recording electrodes that is processed. The potential difference is measured in volts and the amplitude is usually measured in microvolts. Okay, so what happens, the recording at the stage of recording, electrodes will get myoelectric signal. That myoelectric signal is converted into an electrical potential that can be processed by the amplifier. Okay, here the duration of the potential is a measure of time from onset to cessation of the electrical potential. So the phase of the potential is the number of times the wave forms cross or touch the baseline after the onset. Here we can find different types of phases of EMG. One is diphasic and also triphasic. This one is polyphasic. Okay, all these are normal types of potentials which are produced at different phases of EMG. Okay, so diphasic, triphasic and polyphasic. Okay, here this is then so if you see the wave in an diphasic, so here you can obtain this potential and upward and this downward one. So here you can find this baseline. So that means the wave which touches starting from ending of the baseline, that becomes one wave and then the next wave. So it appears as diphasic. So the waveforms may be at different types. One is triphasic and polyphasic. Okay. Yes. Now, what is the other one? That is amplifiers. Okay. So what is the function of this amplifier here? The electrical activity derived from the body is very small and contains undesired signals. Okay, which need to be conditioned and amplified. For this purpose, an amplifier is needed. The amplifier is the part of the instrumentation system that conditions and amplifies the electrical output of the electrodes. Okay. The major principle of use of an amplifier is to amplify the received signal. It also rejects the undesired signals, keeping the desired signal from the motor unit. Okay, that desired signal is going to displayed on the cathode ray oscilloscope. So this one is the cathode ray oscilloscope. In the cathode ray oscilloscope, here you can find, this is the cathode ray oscilloscope. So this one helps or which permits visual display of the motor unit potential. But this oscilloscope does not provide a permanent record. It only allows the signal to be displayed for only a few seconds for visual inspection. And however, if it is connected with any speakers, okay, speakers as well as an computer, we can, here you can see, so we can record the sounds of that motor unit potential as well as the waveform which is displayed can be stored as a data in the computer. Okay, and that data is used for further studies. Okay, so this is about the functioning of the electromyography. Once this motor unit action potential, which represents the electrical activity derived from muscle fibers of a motor unit, so this can be appeared in this way. So here you can find the waveforms. This may be biphasic form. So the motor unit which is appeared here as motor unit action potential appears in this manner. So what happens? And this muscle fibers of a single motor unit sometimes are intermingled in a muscle, they are distributed in time and space. Because of these temporal and spatial properties, the motor unit action potential recorded in the needle examination is a summated action of all potentials of various muscle fibers of the particular unit. Okay, so they have taken, so this motor unit is supplying to different muscle fibers, isn't it? For each muscle fiber we will get some sort of wave, and all these waves are getting summated to get a final wave that is called as motor unit action potential. Okay, so if you see the normal motor unit characteristics, or sometimes the nerve potential of a normal motor unit, which are very small, that is 20 to 200 microvolts in amplitude, and with a short duration of 1 to 3 milliseconds, and which is also having diphasic waveforms with initial negative phase, which are observed in the motor end plate region of the muscle. Okay, an important finding is that the patient gets sharp pain as this potential appears, which disappears when the needle position is changed. Okay, nerve potentials represent discharge from very small nerve branches which are penetrated by the needle electrode. Okay, so and at rest, a normal muscle is electrically silent. Okay, apart from any occasional nerve discharges which are particularly noticeable, if the needle is near to the motor point, then only you can find some changes. Okay, these discharges are initially negative in deflection and of higher frequency than fibrillation potentials. Small negative deflections due to end plate potentials may also be seen occasionally. Okay, so this is the normal waveform, and as the strength of the muscle contraction is voluntarily increased, what happened? Additional motor units are recruited, so that during strong muscle contractions, motor unit potentials get superimposed on each other to obliterate the oscilloscope grid. This situation is called as interference or